Good morning. How are you?
Hi, Katerina. Good morning to you as well. I'm great, thank you.
Great. We will start in around 6 minutes or so. So, yeah,
Yes, I
you
believe
have
so. And now I see that it's available in Google Drive. You put the PDF
yep.
up. I was writing an email to you because I couldn't find it previously, but it's perfect now.
Great. I'm glad that it's working, and yeah, I can hear you well on everything, so that's good too.
Okay, great.
So, yeah, I'll share really quick. I'll mute myself because I will share on Twitter and so on that we are starting, so
Okay,
people know that we're starting.
sure.
Welcome, everyone. Nope, I tried to invite you up. I don't know if... Yeah, there you go. How are you today?
Thank you, Katrina. Awaiting the room. Thank you.
Great. Yeah, we'll start in around 4 minutes. So, if everyone thinks this is something interesting to share, feel free to share the room with friends or people, colleagues, and we'll start in 4 minutes. Thank you. Yeah, hi everyone. We'll start in around 1 minute. Feel free to start checking out the presentation that we will be discussing today, and or share the room with people you think that might be interested in this. And yeah, we're looking forward to discuss this. So, and of course, special thanks to Arpad, who was so generous to come here today.
Well, it is my pleasure to be invited. Thank you.
Okay, how's the weather where you are? Here today is nice. It was raining in New York for a bunch of days, but today is good.
Well, it's in the afternoon. It's nice weather here. I'm from Budapest, Hungary, so it's far away from New York.
Yeah, when the rain and it was getting cold, I was already thinking of all the months that I'll have to be here in the bad weather and figuring out how to escape. No, it'll be all
Well,
good.
in Europe we have an energy crisis, so maybe the whole winter will be very cold,
Yes, yes, I
at
know.
least in the rooms.
Yeah, my parents live in Germany and my brothers, but also my family is from Germany and Portugal. So in Portugal, probably will be fine.
Yeah, weather is relatively nice,
Yeah,
and they are also not exposed to Russian oil as much as we do.
yeah. So in Porto, where I'm from, it's in the north. It gets a little bit cold and very humid. So it's like this cold that gets in your bones. And the houses are of granite stone, so it's really humid and cold. But usually people have like fireplaces or this ovens.
Yeah, that's cozy.
Oven. Yeah. So yeah, I think we can start. So welcome everyone to Science Society, and a special welcome to Arpad today. And before we start, let me give you a short introduction so you get to know him a little bit. Arpad Dobolyi received his university degree in chemistry at the Eötvös University, Budapest, Hungary. And he obtained then his PhD at the Semmelweis Medical University in Budapest in cellular and molecular physiology. And he then became a postdoc of Dr. Ted Ustin at the National Institute of Mental Health in Vesta. And he returned to Hungary in 2005 as a Marie Curie Fellow. And he worked in the lab of Professor Miklos Palkovits. And then since 2007, he is a research associate professor at the Semmelweis University. And in 2017, he became a full professor. And in 2018, the head of the Department of Physiology and Neurobiology at the Eötvös University. He supervised 10 postgraduate fellows, and he obtained, and they all obtained their PhD degree, and 4 postdoctoral fellows. And his current research, he's mostly interested at the neural mechanisms controlling social behaviors, and he has a really high H index too. So it's an honor having you here, that you took, the time out of your busy life to come here and talk with us. And usually before we start, we ask a couple of interview questions to get to know you a little bit better. So was it always something you wanted to do, to become a scientist, or was it something that came later on, or maybe a class you took or a book you read? How did you choose this life?
Yeah. Well, thank you very much for the nice introduction. Yeah, as for the question, yes, it was an easy decision for me because I always wanted to be a scientist. What was difficult, what kind of science I'm really interested in. Uh, so in high school, I was mostly focusing on physics and chemistry. As you heard, even my university degree is chemistry after all, and I took many courses in biology, of course, and physics. But finally, I realized that neuroscience is the one I am really interested in. So within neuroscience, I also had different ways. During my PhD, I did neurochemistry, and then at NIH molecular biology, then a lot of histology, and really when I just started my own lab, that's when behavioral aspects came in, and social behavior is indeed my recent interest, and I would say my current decision. So early on, during high school, I was already I decided to become a scientist. I went to a special school for natural sciences education, and somehow I don't remember. Before being wanted to be a scientist at a very young age, I wanted to be a soccer player, of course.
Well, like every
Like every
European boy.
indeed.
So,
and when my teacher asked me, but that's not a profession, say something else, then I wanted to, then I a trainer, a soccer trainer. But that was early on, and then science came in.
well, a trainer, probably you can use a lot of your research to make a proper training behavior plan for people, maybe.
Yeah,
So
that's
maybe in the future, maybe scientists will become trainers.
yeah,
So
that's at some point when you go on with your profession and young people join the laboratory, it's indeed a similar activity.
yeah, and you touched a little bit upon how you also then chose this field of research. It's interesting how you transitioned from physics to neuroscience and behavior. Um, so, was that a curiosity of yours? Like, how do people behave? Because you first started with physics, and the second question is maybe how this project came about. Then out of that curiosity, maybe.
Yeah, at some point I just felt it's really the most exciting thing to learn how we human work, how we function. So at that point it suddenly became obvious that's what I'm interested in. I mean, you being a scientist, you are really lots of freedom. So that that's no matter what you do, it's I come in work in the morning, and I can do what I want to, more or less. Well, I have to duties, but so that's for science, and somehow neuroscience. Somehow it was obvious, and then when social neuroscience came in, I did too much molecular stuff and histology, and I just felt they are too far away from you know from exciting things about us. And then that's how social behavior came in. Also, in my talk, I will tell a little more detail about how molecular discoveries led to actual behavioral studies. Um, so that it was not like a forced out thing from my part, but also it was I feel it was a natural development of the science as I built it up gradually.
Interesting. And for physiology, you need a lot of physics, so It's
Yeah,
actually
that was useful. Yeah,
yeah.
during my studies and later on as well. That that's right. That's right. You know, yeah, after all, different parts of science are always connected if you can understand the connection. So that's somehow it's also true. But it also requires some kind of boldness to enter into a new field, like you know, when to start doing behavior when your education was really not behavior. That's a, you know, big decision because it could lead to failure. But I, I I seem to be lucky so far. At least my students can do it very well. So it's great.
Yeah,
No?
I'm glad you did, and I yeah, I kind of feel it's maybe more and more important to like not be a very specialist, but that's a whole other discussion. You know,
Yeah.
the AI coming up, maybe we need to be more general than we were in between. So, but yeah, the stage is yours for your presentation. Please, everyone, click on the link that's pinned on top of the room. There's Arpad's presentation to follow along, and the stage is yours. Thank you.
Yeah, thank you very much, and I welcome everyone who showed up to listen to this talk. The title is a thalamo-preoptic pathway promoting social contact behavior. So preoptic area is the anterior part of the hypothalamus. In that sense, it is a fairly rare thalamo-hypothalamic pathway. There are not too much connection between these two diencephalic structures. Anyway, my university is shown up upper right corner. It's very nicely located, and the paper why I was invited here recently appeared in Current Biology, and the first author was David Keller, who's actually a medical doctor doing a PhD in my lab. So if you go to the second slide, the story, it will not be a very long introduction, but still it started 20 years ago when Ted Asdin at NIH discovered the novel neuropeptide. That's parathyroid hormone 2. That time we called it tuberoin fundibular peptide 39. He purified it as a peptide from the hypothalamus as the ligand of the parathyroid hormone 2 receptor. So that's when I joined the lab and described that PTH2 is coded by a single gene which does not code any other peptide. I also showed that PTH2 neuropeptide is expressed in the posterior intralaminar thalamic nucleus. That's not very well known thalamic nucleus. It's shown on the right. Here you can see this bluish color are PTH2 neurons, and the nucleus itself is located ventromedial to the medial geniculate body and just lateral to the white matter of the medial lemniscus above the substantia nigra. Later on, we showed that this peptide is induced in mothers who are taking care of their pups. So the third slide shows the peptide, the mRNA level of the peptide, upper panel, and the lower panel is the peptide level, which is very low in control females, high in mothers. But if we take away the pups from the mother, it becomes low again. So based on these expressional data, we did lots of functional studies on the maternal function of this peptide, including one example which is shown here, which demonstrated that the preoptic area parathyroid hormone 2 increases maternal motivation. So this is a pup-induced place preference. If a mother used to be together with the pups in one cage, and later on she has the option to choose between two cages, she will stay more in the cage. Even if the pups are not there, she will stay more there. That's a pup-induced preference. and a PTH2 parathyroid hormone 2 receptor antagonist can eliminate this aspect of maternal motivation. It was recently, 2 years ago, when another group in Germany published that parathyroid hormone 2 is induced in zebrafish if the zebrafish is kept socially. So that means in isolated zebrafish, the level is very low, but when many fish are close to each other, this peptide is induced. And when they are separated again, it goes back to low level. On the left, this is now slide number 5. On the left, you can see isolated zebrafish brain, and lower panel, social left, peptide level, right mRNA level. The position of that was actually corresponding to the posterior intralaminar thalamic nucleus. And this research group also established that it's a mechanical input which induces the peptide through the lateral organ, which feels the vibration of water elicited by other zebrafish. So we after that we became interested in adult social interactions, and first we looked at the activation of the posterior interlaminar thalamic nucleus, or PIL, in social situations. Here on slide 6, you can see these black dots. These are CFOS positive neurons. So this is a technique which can histologically visualize the active neurons. On the left, this is a female which was in contact with another female, and on the right is a control without reunion. If they were reunited but they were separated physically with a cage or mesh, then induction was not found. Based on that, we formed the hypothesis that these PIL neurons somehow control social contact behaviors or some aspects of social behavior. To functionally address that, we had to choose some sort of behavior. There are different types of direct social contact behaviors, as shown in slide number 7. Some of them are reproductive, like nursing or copulation, sexual behaviors, could be aggressive, but there are also positive valence behaviors, such as social grooming. In a monkey group is shown on the right. Social grooming is important to keep together the group in this species. So in the first experiments I speak about, presented in the paper, it's always friendly interaction between adult female rats was examined. Now we started other type of behaviors as well. To functionally address the role of these neurons, we use the chemogenetics. So stimulatory, inhibitory, and control virus was injected into the area of the pill. That's slide number 8, what I speak about now. On the right, you can see the position of an injection site, where M-cherry, the fluorescent protein, is visualized here with DAB immunolabeling. The experiment was done that the operation, the injection of the virus takes place. We have to wait about 2 weeks while the animals recover, and the virus expresses the stimulatory dread receptors. These are artificial receptors with which we can stimulate the infected cells. And the social behavioral experiment is done in a way that the animals are separated just for an hour to increase subsequent activity, and then they are videotyped for 10 minutes. Next slide shows a validation of our chemogenetics with the stimulatory virus. We injected CNO, close-up in N-oxide, to stimulate the receptor, and then we found CFOS activation in 90% of the neurons expressing the stimulatory DREAD receptor. So that demonstrated that indeed these cells are stimulated in the experimental situation. During the 10 minutes direct social behavior, when they were placed in the same arena, two animals, so again two female animals, slide number 10 shows the 6 different elements of social behavior which we looked at. One of them is anogenital sniffing, non-anogenital sniffing, chasing, social grooming. Social grooming means when the forelimbs of one of the animals touches the body surface of the other, then mounting and passive social interaction, kind of side-to-side contact. So here, next slide 11, you can see the results. The first column is always a control injection, and the third column is also a control injection, like 3 or 4 days later. But here the yellow column is the CNO injection when the receptor is activated, either stimulating the cells or inhibiting them. This shows the effect of stimulation. As you can see, social grooming was the specific social behavior which was increased. Non-anogenital sniffing had a tendency of reduction, but as you will see, it always happened in subsequent days. So social grooming was specifically affected by these neurons. On the right, you can see social grooming in a way that same animal data are connected with each other, so that you can see the result of individual animals. When inhibition was applied, social grooming changed the opposite direction. So there was a reduction in the amount of social grooming these rats exhibited. And we also applied the control virus, where CNO injection was without effect. This is slide 13. Slide 14 shows some control experiments. You know, we were just generally interested if these animals are okay. For example, this is an open field test where the total distance is shown, which was not affected by either stimulation, inhibition, or control, suggesting that the locomotor activity is not affected. Similarly, their anxiety level was also not changed, for example. Next experiment, we decided to investigate not all the neurons of the PIL, but only those neurons which were previously activated by social interaction. So these socially tagged neurons were made to express Cre recombinase using a virally induced V-gate TETON system. Now this is slide 15. This V-gate system was previously published in Neuron by my collaboration partner Valery Grinevich at Heidelberg University. So in this case, an antibiotics, doxycycline, opens a time window during which social interaction takes place between the animals, which expresses Cre recombinase in those neurons active in that situation. And later on, Cre-dependent expression of the DREAD either stimulatory or inhibitory or control is performed, as you can see in the lower right panel of slide 15. The next slide, 16, shows the results of these socially tagged neurons. Upper line, you can see the effect of stimulating the stimulating those neurons in the pill which were previously active in social situation, and you can see an increase in the amount of social grooming behavior, but no change in most other. Again, non-genital sniffing always shows this reduction. So inhibition of these neurons led to a decrease of social grooming behavior. And again, next to the column table, you can see individual animal data connected with each other in the control day, the CNO day performed 1 day later, and the second control day to check if the amount of behavior goes back to baseline level. When no contact was allowed between the animals, that's shown on the right of slide 16, so separated with a cage, then the amount of time spent around the other animal was not affected by either stimulation or inhibition of socially tagged PIL neurons. Okay, so now this established the function of the neurons of the PIL, this posterior intralaminar thalamic nucleus, but we also wanted to look at where do these cells project. So we injected an anterograde tracer, biotinylated dextran amine, into the nucleus. Slide 17, upper right panel, you see the injection site, and upper panel in the middle, you see the summary. As for the cerebral cortex, only the medial prefrontal cortex, mostly the limbic cortex, was an area where these cells project. So considering that they are a thalamic nucleus, it's not some cortical projection, but stronger projections were sent to the lateral septum. Also, third panel in the middle, medial preoptic area. Here you can see the third ventricle on the right of this panel, optic tract, and high density of anterograde relabeled fibers in the medial preoptic area. Paraventricular hypothalamic nucleus had some fibers. Medial amygdala had strong labeling. Dorsomedial hypothalamic nucleus, periaqueductal gray, and the lateral parabrachial nucleus. So that's the projection pattern of these neurons. We investigated next which of these target area of the pill is activated in response to social interaction with another animal. And in fact, all of them showed some activation. What I'm telling you is slide 18. However, when the two animals were separated physically so that direct physical contact is not allowed, then the activity level in most of this brain area decreased, but there was one area, the medial preoptic area, where there was no activation at all when the animals were not allowed to directly interact with each other and thereby exerting somatosensory stimulation of each other, kind of social somatosensory stimulation by direct contact. That's why we decided to specially investigate the function of this pathway from the pill to the preoptic area of the hypothalamus. Functional investigation of the pathway took place in two different ways. First, we injected retrogradely spreading virus. This is now slide 19. So we injected this virus into the medial preoptic area. This spread retrogradely all the way back to the cell bodies of these cells into the posterior intralaminar thalamic nucleus and possibly other brain regions as well. However, a second virus, a CRE-dependent virus, this DO is the double inverted operating frame, which is makes it CRE-dependent expression. It was injected into the PIL, so it's a double viral injection. And when these cells of the PIL projecting to the preoptic area were inhibited, then as you can see on the right, the amount of social grooming behavior was markedly reduced. The second approach to investigate the function of the PIL preoptic pathway was that the injection of the ligand, the artificial ligand of the DREAD receptor, was not injected intraperitoneally, that means systemically, but rather directly into the medial preoptic area. And thereby other projections from the PIL, which are not in contact with CNO, are not activated or stimulated. So in response to local injection of CNO into the medial preoptic area using stimulatory receptors, now this is slide 20, there was an increase in the amount of social grooming behavior. So the conclusion of these studies is that the PIL preoptic area pathway controls social grooming behavior in the rat. As a summary of our findings from so far is that social contact activates a number of neurons in the PIL. Chemogenetic stimulation of these neurons increases friendly social contact, while inhibition has the opposite effect. The target areas of the PIL show CFOS activation in response to social interaction, especially the preoptic area, which is not activated unless direct interaction is allowed. And the stimulation of the pathway itself, the PIL preoptic pathway, leads to increased friendly social contact, which is the grooming behavior. But we had another question. What is the role of the neuropeptide parathyroid hormone 2 in the functionally characterized thalamic preoptic pathway? So we did additional studies using real-time PCR. We demonstrated, this is now slide 22, we demonstrated that the expression level of the neuropeptide is increased if the rats are not kept isolated, but they are kept together with other conspecifics. So in that sense, we kind of found the same thing as was previously reported in the zebrafish, and actually a similar finding which was found in mothers when their pups were present. Next, we investigated PTH2 peptide in the preoptic area. Are they present in those terminals? The answer is yes. Here, slide 23, green shows the PTH2 containing fiber terminals, and retrograde tracer, cholerotoxin beta subunit or CTB, was injected here. Then we found lots of double labeled cells in the PIL, suggesting that the origin of these PTH2 fibers in the preoptic area is the posterior intralaminar thalamic nucleus. Is the receptor of PTH2 present in the preoptic area? Actually, we already previously described the distribution of PTH2 receptor using a transgenic approach, and indeed the receptor of PTH2 neuropeptide, the PTH2 receptor, is present in the preoptic area in a distribution similar to the distribution of PTH2 containing neuronal terminals. Next, shown in slide 25, we demonstrated that PTH2 receptor expressing neurons are CFOS activated in response to social interaction. So on the left, the red color is the PTH2 receptor. This is a transgenic animal crossing a PTH2 receptor Cree animal with a reporter mice, and these and the red is CFOS, and these greenish dots on top of the CFOS indicates double-labeled cells, which is shown by the white arrowheads. And indeed, the number of double-labeled cells increases in response to social exposure. Most importantly, as far as function of the PTH2 neuropeptide in the preoptic area, an antagonist of the receptor was injected into these rats, into the lateral ventricle of the rats, and the behavioral effect of PTH2 receptor antagonist were investigated. The same social behavioral elements were evaluated as previously, and as you can see on slide 26, on the right, social grooming behavior was significantly reduced in response to PTH2 receptor antagonist injection as opposed to a vehicle ACSF artificial cerebrospinal fluid injection. So the conclusion is that not only the thalamo-preoptic pathway, but its neuropeptide neurotransmitter PTH2 also contributes to social grooming behavior. What could be the target of PTH2 containing terminals in the preoptic area? That's what we addressed in the next experiment shown on slide 27. This is actually not in this now published paper, but it was done previously. The targets are actually GABAergic neurons. In the middle panel, they are green color. Vesicular GABA transporter Cree animals were mated with the reporter mice to make GABAergic cell bodies visible, which express CFOS. So the CFOS cells, which are activated in response to social interaction, are almost all inhibitory GABAergic neurons. And on the left, you can see that the CFOS expressing cells are surrounded by PTH2 containing fiber terminals. In fact, a subset of GABAergic neuron in this area are the galanin neurons. It's a neuropeptide, and the double labeling with parathyroid hormone 2 and galanin in a correlated light and electron microscopic study demonstrated the presence of synapses. On the right, you can see electron microscopically a synapse based on the presence of synaptic vesicles, synaptic densities, and so on. So there is a direct real synaptic contact between the terminals and these neurons. Does PTH2 neuropeptide affect these cells? It was directly investigated by patch clamp electrophysiology using preoptic slices. A whole cell patch clamp was done, and the peptide was just injected on top of the cell we recorded. There was a concentration dependent increase in the frequency of the firing rate of GABAergic cells. So these were all GABAergic neurons. If an antagonist, the PTH2 receptor antagonist, was injected right before the neuropeptide, as you can see on slide 28, left lower panel, then the antagonist kind of prevented the excitatory action of parathyroid hormone 2 neuropeptide. We really don't know what is then the function of these GABAergic neurons in social behavior in the preoptic area. We did a previous study examining their function in maternal behavior, and that's what is shown in slide 29. When stimulation of these GABAergic neurons increases maternal care and maternal motivation, inhibition reduces maternal motivation. So slide 30 shows a kind of summary. There is an ascending pathway from the spinal cord, from sensory input from nipples in mothers or body surface in adult situation, which excites PTH2 neurons in the PIL, which in turn projects to the preoptic area, including galanin neurons there, to promote social grooming, maternal motivation. It actually, I didn't show data, but it also project to the oxytocin neurons in the PVN. And there are other projections, amygdala, lateral septum, medial, prefrontal cortex, whose function is unknown at present. The next question was if this pathway is present in the human brain or not. So slide 31 shows human brain. The drawing on the left, there is a framed area which is shown in the middle panel, which is a Nissl staining, and the PIL is indicated by black dots, the border of the PIL indicated by black dots here. And on the right panel, this is a Luxol fast blue staining, which labels myelinated fibers. So the medial lemniscus is immediately lateral to the PIL in the human, just as it was in the rat. For some neurochemical characterization of these PIL neurons, this is a rat study. We double labeled it with carbindin and established that most of the PTH neurons are carbindin positive, but we also demonstrated that the carbindin cells are not GABAergic. That means these are the PTH2 cells, which are carbindin neurons in the PIL, are excitatory. In studies I do not show here, we demonstrated the presence of glutamate in them, by the way. Here, the carbindin immunostaining is shown in the human brain. The panel in the middle shows carbindin immunostaining. The framed area is shown on the right, which where you can see individually labeled neurons within the PIL, but not outside of the PIL. And finally, the PTH2 receptor was demonstrated to be present in the human medial preoptic area, as shown in slide 34. Drawing on the left, low magnification picture in the middle, and higher magnifications on the right. Lower panel on the right shows immunolabeled cells. Finally, the last experiment I show you is about the inputs to the pill. We injected retrograde tracer, cholerotoxin beta subunit, to an area identified by the presence of PTH2 neurons. We identified on retrograde labeled cell bodies in different levels of the spinal cord, also several neurons in the cuneate and gracile nuclei, but also in the external cortex of the inferior colliculus and the hypothalamic ventromedial nucleus, as shown on slide 35. So in summary, as the last figure, I show you the standard textbook pathway which delivers tactile information, mechanical input on the surface of the skin towards the primary somatosensory cortex. So this is the dorsal column medial lemniscal pathway, which relays in the ventro-posterolateral thalamic nuclei, VPL. Our data suggest that there are collaterals terminating in PIL, and specifically social information may be mediated by the PIL as well, projecting to other brain area, including the preoptic area. And because this pathway, shown in blue on slide 36, bypasses the cortex, this information probably will not become conscious. So it affects our instinctive behaviors um, without being becoming conscious in the cerebral cortical processing. And finally, I would like to acknowledge all my people in the laboratory, postdocs and PhD students, collaborators, and support as well. And thank you very much for your attention. And if you have questions, I'm very happy to try to answer them. Thank you very much.
Well, thank you for sharing this really amazing story of research and social behavior. It's really it's so much work, and you covered really all different aspects of the story. So it's really wonderful work. So thank you for sharing it with us. And yeah, the question that probably you got probably asked a lot of times is, are you thinking of looking at differences maybe of human tissue, postmortem, if in people with autism or other, social behavior differences show like less expression or maybe less neurons of the social behavior pathway? Thank you.
Yeah, I mean the human aspect, the human consequences of these results is indeed huge. There are many neuropsychological disorders where social behavior is affected, and indeed um, um, autism is it has different forms, but still physical contact is often not well, it's affected for those people. Certainly, these data could have implications there, and I hope it will be investigated. As far as investigating postmortem tissue, we are trying to get postmortem brains, but it's not very easy at all to get postmortem sample. And even though I'm in Budapest, there is a brain bank with which I'm connected. In that particular brain bank, we do not have appropriate postmortem brains. so I hope it will be done by someone who has proper access to the postmortem tissue. As far as additional approach could be some sort of pharmacological approaches, and I'm really hoping that pharmaceutical companies will be interested in the PTH2 receptor and try to develop small molecules acting through this receptor. But as you may know, neuropeptide receptors are not an easy target as far as small molecules.
Yes, definitely. So would it? Did you, um, you know, did you maybe see differences? Probably not. There's probably no genetic. Is there a genetic background, maybe difference of levels of expression that one could detect, in tissue collected? Probably
Uh,
in the brain itself, but ideally it would be a general, more general.
Yeah, we actually just started to investigate models, animal models of autism spectrum disorder. There are indeed genetic models where the lack of some protein makes these animals good candidate as being a model of this disorder. In addition, there is the pharmacological. It's also possible to pharmacologically induce that kind of properties. So right now, um, we we are looking at those models, whether this pathway or the peptide or any component of the system may be affected or not. But right now, I do not have data to present on that.
Yeah, and one last question before everyone else gets the turn. So the GABAergic cells are involved too. So did you maybe try to give neuromodulators or agonists or that would activate those GABAergic cells, and would that lead to increased grooming behavior, social behavior?
Yes, that's a good question. What we are trying to do is kind of repeating the chemogenetic study on the preoptic GABAergic neurons, which I showed as far as maternal behavior, but repeating that to investigate adult social behavior. As far as pharmacology on GABA receptors, they really are all over the brain. So there may be some local injection, but even if we locally inject these GABAergic neurons in the preoptic area are actually many of them, most of them, I would say, are projection neurons. So they act in many different brain sites where they project, um, and the pharmacological treatment of based on GABA receptors, where there are so many drugs on anxiety and other which act on GABA receptors. Of course, anxiety is a critical issue as far as social behavior, because whenever the animals are not relaxed, they reduce significantly reduce social behavior. So certainly the two systems are connected, but it's not easy to specifically investigate the function of these GABAergic neurons in the preoptic area with GABAergic pharmacological tools.
Yeah, I agree. It's a very it's a broadly. I was just thinking for post partum depression, if we would find a acute drug, even if be larger scale, if it would enable for those neurons to express more of that protein, then maybe would it would maybe solve something. But yeah, I agree. It's very large scale. And then last, I said last question, but one is, did you see any immune system component that kind of, or are you planning to look for immune system component that could modulate the circuitry? Just because there was this paper in Nature from 2016 where they saw an unexpected role, the title is Unexpected Role of Interferon Gamma in Regulating Neural Connectivity and Social Behavior. And I was really puzzled when that paper came out. And when I read your work, it kind of reminded me again of that paper, if there could be maybe immune system component that could modulate this circuitry.
It's absolutely possible that it does. Um, our approaches were chemogenetics, which is not a molecular level investigation, but rather network level. In addition, as far as molecules, we focused on the neuropeptide. So that approach clearly leaves open the any possibility of being affected by neuroactive substances, including cytokines, in the preoptic area. So it's absolutely possible. Indeed, we are actually started to do RNA sequencing in the preoptic area, comparing socially isolated and group housed animals, but we have no data yet. So I don't have molecular information, but absolutely possible that the two systems are affecting each other.
Very interesting. Thank you. Frank, Joyce, Nope, did you have questions? Or anyone in the audience, please raise your hand if you have a question. Thank you.
Yeah, this is Frank. Thank you very much, Arpad, for this amazing study and the presentation. I just wanted to kind of clear the exact setup on slide 5, 7, and 10, where you kind of relate certain the the behaviors to each other in the setup, and I didn't quite well understand why this PTH2 is higher on the slide 5 on the isolated plus the short social interaction, and then I wanted to know this: where's the difference with this PTH2 with the social grooming and vice versa, this aggressive fighting? What's the difference there? I mean, an aggressive fighting situation like it is pictured there, I mean, it is a social interaction, at least. So I just want to be very clear to understand what is in the setup and what did you find with that. Thank you.
Yeah, yes, yeah. Thank you for the question. So slide 5 shows a previous data by another research group in zebrafish. That was like the first data that the PTH2 could be involved in somehow in social interaction of animals. Here they showed that the level of PTH2 is low when fish is isolated, but even a short social interaction induces, that means short in time, so a brief interaction already induces PTH2 levels. And when the animals are kept socially, that means many fish together in the same tank, but we will isolate them, then a short isolation will lead to the reduction of PTH2. So that was their finding in the zebrafish. And when we demonstrated later on that PTH2 level is also induced in the PIL in rat. It was a simple experiment, isolated for 1 week or kept together constantly, and that's what they were compared. So the conclusion from that can not be more than what the experiment was about. Not a very good time resolution what we did. So then slide 7. It was an introductory slide to tell you what kind of social behaviors include direct interaction and reproduction aggressive, both indeed, but the experiment we did so far was only one of them. That was a positive valence interaction between friendly female animals. Most of the time they were actual siblings growing up together, separated briefly before the experiment so that they are more interested in each other when they are returned to the cage. But I believe investigating the function of this thalamo-preoptic pathway in aggressive behaviors, like using male rats, for example, defending its cage against an intruder, that's a suitable model to investigate aggressive behavior, or reproductive behavior, such as sexual behavior between male and female rats, and manipulating this pathway. uh, I believe it's realistic that the pathway does play a role in aggressive behavior or sexual behavior, and it's certainly something we will look at in the future.
Amazing. So, last question from my side. Do you think there's any effect of families onto this? So, is there? Do you think that there's a difference if let's say father and daughter, right, or the grandparents and grandchildren, that social behavior between those family structures is representing more of these PTH2, or does it matter at all compared to larger social systems like country identity and all of that?
Yeah, so that's a very good question. In the fish, the peptide kind of just measures the density of the population. In rat, it of course could, or in rodent, human, it could be very different. What is the function of this social grooming between friendly animals? One possible function is, for example, consolation behavior, and it was shown not in the rat but in wolves, um, uh, that Larry Young, for example, showed that consolation behavior uh, takes place between siblings, between mates, if it's a monogamous wolf species, but not in just two stranger animals. So it's a possibility in the rat too, that family ties has an influence on their behavior, and the the role of this pathway somehow is involved in that. But right now there are no data on that.
Well, thank you very much.
Thank
Yeah,
you.
just to comment on that, do you look at, or are you planning to look at epigenetic, like methylation levels, maybe differences? It's a really interesting question. And transgenerational inheritance,
Yes,
maybe.
indeed, the transgenerational inheritance is one thing why epigenetics is interesting here. Actually, also when mothers are investigated, whether the first pregnancy and second and third pregnancy, there might be differences between them, how they take care of the pups, and that could also be epigenetics. Um, Clearly, social interactions too. or even neglected a child when grows up, it could be epigenetics involved. So that's a very exciting story, but we are not investigating epigenetics right now.
Yeah, I have one additional question there. What for neuroscience? What's the difference in the challenges when you research animals and compared to humans? And I'm not talking about the availability of that. I mean, if let's just assume you would have that available. I'm interested into the difference in the challenges.
Uh, well, working with human, because my background is not a medical doctor. I really um, so well it's so working in human in that sense more responsibility. Although in animals you have to keep all the ethical rules and work. You really have to be certain what can be done and what cannot be done in animals, but still, because invasive approaches are possible, like all these chemogenetics approaches, certainly this is not possible in human. Many of the psychological aspects, on the other hand, can be better examined in human. When we compare the two species, I would say the social behavior of human is is still dominated by the cerebral cortex processes because of the growth of that area, and there could be many other processes in social behavior such as mentalization, empathy, and so on, which build up. But I still believe that this pathway, which is plays an important role in rodents, uh, is still there in human, and it still has a role and affects behavior despite the the huge development of and the dominance of the cerebral cortex in defining several aspects of social behavior. Some instinctive behaviors are still there, and this pathway could be involved in human too.
Okay. I have a question. This is Joyce. It's all
Yes.
very interesting. And I was just wondering if, there's a lot of research on the relationship of autism to the microbiome and to diet and so on. And if I'm wondering if you see any connection in with your research and this train of research. Anyway, thanks.
It could very well be connection in the future, but right now, the microbiome of these experimental animals was like not controlled, um, and uh, that does not make it possible to make a connection. So I say it should be investigated in the future, but right now there is no data here.
Okay, thank you. Also, the parathyroid hormone is very involved in things like calcium metabolism. I don't know, I missed some of the talk. Does your work have any relationship to that?
Yes, that's indeed a basic question. So parathyroid hormone and parathyroid hormone related peptide, they are acting on the parathyroid hormone 1 receptor, and they are not acting on the parathyroid hormone 2 receptor. And this peptide, the parathyroid hormone 2, it does act only on the parathyroid hormone 2 receptor and not at all on the parathyroid hormone 1 receptor. When we looked at the expression of the PTH2 peptide, it is found in very low level in a few organs. such as the kidney, but not in bones. It's really abundant in the brain. Actually, only 3 sites within the brain. One of them is the PIL I was speaking about, and 2 other similarly restricted brain area. And as far as we know, PTH, the famous hormone, of course, in calcium metabolism, it does not play any role in brain function. So its receptor is not there, the peptide is not there. So in the brain, PTH2 is, and in the calcium metabolism, well, we did lots of studies whether PTH2 itself can be a hormone or not, but right now there is no evidence it would be a hormone. There is no evidence it's present in the blood, and also there is no evidence that it has any effect on calcium metabolism.
Okay, thank you.
Hi, LT, please go ahead. Welcome.
Thank you. I really enjoyed this talk. Question: when you mentioned that PTH2 has no what do you call that function right now, right? But the PTH2, I remember I'm looking at your slides, also following your talk. You said it's only making one message. Is that what you're saying? So there's no other form of a PTH2? How similar?
Yes.
Yes. How similar is PTH to PTH2? That's my question. Thank you.
So if I understood correctly, you ask about other peptides similar
Yes.
to PTH2,
Right.
and many of the genes which encode neuropeptides, they often encode more than one neuropeptide, and they are, translational cleavage leads then to the appearance of actual neuropeptides. We found no evidence at all that the gene of encoding PTH2 would encode any other neuropeptide. So it seems like it's a kind of exception among neuropeptides, that one gene encodes only one peptide, and this peptide is not encoded by other genes. Sometimes related peptides are encoded by other genes too, but in this case, in that sense, it's a pure system molecular biologically, and also pharmacologically, it's a simple system, one peptide, one receptor. There is actually very little amino acid, or maybe not at all, I would say. There is no sequence similarity between PTH and PTH2. They have the same name because there is sequence similarity between the receptors. PTH1 receptor and PTH2 receptor has like an almost 50% sequence similarity, but the peptides, they do not have sequence similarities at all. Their three-dimensional structure resembles each other to some degree, but still, there is no other peptide to which PTH2 would be more similar than PTH1. So essentially, there is no similar peptide.
Okay, so you already hedging ahead of my next question. So because if the receptor is very similar and then structure-wise they're similar, so is it possible that they could be mistaken? Could that be used? Like do you see what I'm? Hold on just a second. That you would target you would bind it to the wrong receptor. Thank you very much. Have you seen evidence of that?
So I'm sorry, I couldn't understand the question.
Hold on. Can you hear me all right? So
yes, I can hear you.
Okay, because the naming is PTH and PTH2 because their receptors are similar. Okay, so in this line of thinking, I'm asking, have you seen evidence of PTH2 bound to the receptor specifically known up to now for PTH? That's my next question. Thank you. Is it clear now?
Yes, so whether PTH and PTH2 bind to to the receptors mutually?
Yes.
That's what you asked.
Yes.
So so actually PTH2 was a new, it's a new name. When we named it, we called it tuberoinfundibular peptide based on its presence in the hypothalamus. But this database is kind of forced out the name PTH2, which now makes it very similar to PTH1, the actual hormone. There were detailed pharmacological investigations, including not only these two peptides, but their rat version, mouse version. Actually, mouse and rat are exactly the same amino acid sequence, but the human PTH2 differs in 4 amino acids, 4 out of 39. So it's actually kind of relatively a big difference. Also, the bovine PTH2 is exactly the same as the human. So there are many possibilities for pharmacological investigations, whether like the mouse peptide binds to the human receptor and vice versa. But still, generally speaking, it's always established that PTH2 does not bind at all to any species of PTH2, does not bind to the PTH1 receptor of any species.
Okay. So it's clear. It's absolutely
in
so
that
far.
sense,
Okay.
it's clear.
Thank you.
Yes.
Yeah. Because I think Kathleen asked a similar question about the immune response. If the receptor is binding that, my head is exploding. how would that? Okay. And what about the localization? Would that make them kind of more accessible? And then when you have, say, 4 mutations are different, that means that, I'm sure people at your lab, other labs, probably doing the knock in, knock out. So there might be ways of using culture neurons or hold on, I'll stop asking questions. Thank you.
Yeah. So indeed, PTH2 neuropeptide and receptor knockout animals are being developed, and they are recently available, and we actually investigating them as far as social behavior. It's certainly all those people who have access to it. It's certainly an interesting next experimental plan for us. Yes.
Okay, thank you. So everybody upstairs, downstairs, I will move myself down. Thank you. Small.
Thank you.
Yeah, thank you so much for answering all these questions. I don't know if anyone has a last question, maybe, because we've been going over an hour, so probably Arpad needs to go back to his work and life. And
Well, it was really my pleasure. I'm happy about the numerous questions.
yeah, thank you, and I really appreciate you spending the time here. I think it's such interesting work, and it opens up so many more questions and projects to be answered, which is always the most interesting type of project. So it's really wonderful work, and yeah, I'm curious to follow your research along in the future, to see what if some of these questions, how the answers will look like. So it will be really interesting.
Yeah, exciting. Thank you very much.
Yeah, and yeah, if there is maybe news updates, feel always invited to come back, and we will be always curious to learn about what is going on in your lab, basically. So yeah, thank you for this.
Yeah. Thank you very much. Thank you for the invitation and for all the people who showed up. I appreciate all the questions as well. Thank you. Bye-bye.
Yeah, thank you, everybody.
Yeah, and thanks everyone for coming, asking questions, and being here. If you like discussions like this, we will have actually this week a few more guest speakers coming. Tomorrow we'll have Dr. Bogosian, who is making bioelectricity in so he makes living photovoltaics. And in the afternoon, Dr. Liu and his colleague will be coming from the British Museum talking about ancient Chinese metallurgy, how it was way more complex than we thought, the composition of creating metals. I think it was really interesting. And then on Saturday, we'll have Dr. Wu coming talking about new wireless optogenetics and photopharmacology technology development. So follow the club if you like discussions like this. And again, Arpad, thank you so much. This was a great honor to have you here, and this research is so interesting and I think also very important for the future. So thank you.
Thank you. Bye-bye.
Okay, I'll close the room in 3, 2, 1. Bye, everyone. Thank you.